Every organ section has two descriptive modes, organ parameters and organ metabolism. In each particular case, these modes will feature particular quantitative values of relative indices, mode menu structure remaining the same for all organs. Once organ parameters mode is selected from the main menu, a hierarchical database structure will appear, containing all the selected organs' parameters used by the simulator in its work. Let us demonstrate such a structure, or a search tree, with lungs taken as an example. First, we select lungs by moving the pointer to the corresponding image and left-click to confirm the choice. Selected organ-related modes appear in the Mode menu. Then we left-click Organ Parameters. Now we can retrieve information we need, like, for instance, doing a sequential search for data concerning content of vitamins in extracellular fluid within lungs. Left-click on Extracell, then on Biochemistry, then on Vitamins. Now we've got what we've been looking for, and that's how we work with all this search tree. Every index engaged in the system is within a statistical normal range. Values of all indices are of common knowledge and can be verified through any reference source. Once organ metabolism is selected, there will be presented all chains of biochemical transformations of different ingredients in a specific organ, provided the organ is selected on the on-screen simulator, as well as all subsequent biochemical transformations of derived substrates, including the Krebs cycle and the ornithine cycle, the energy accumulation processes in the cells of the organism, and the output of the final irreversible breakdown products. Let's see what happens when such mode is initiated for the liver, for example. We'll have to repeat the organ and mode selection procedure. Here and further, for users' convenience, we will be using different colors in the names of the corresponding substances. Highlighted in yellow are the key substances presented in the full cycle of transformations. For example, by left-clicking the yellow rectangle labeled glucose, one will get a complete graph of biochemical transformations of this substance in the organism, here in the liver. To obtain a real-time quantitative value of any key substance in a given scheme of transformations, left-click the yellow rectangle with the name of a particular substance. As a result, the chemical formula of the substance and its quantitative value at a given time will be presented. For example, the quantity of glycogen. Red color marks the beginning or the end of a chain of transformations. Blue color highlights the substances that are attached or detached from the key substances directly in the course of a corresponding reaction. Quantitative values are not displayed in this case. Pink color highlights intermediate substances formed in the process of metabolism, which have not yet completed their biochemical transformations in this scheme and can be used in other schemes. Quantitative values are not displayed in this case. Green color highlights the substances consumed in the process of metabolism. Quantitative values are not displayed. Dark blue color highlights the names of specific parts of the scheme of transformations. All metabolites transformations from one to another are regulated by relevant enzyme systems, indicated in all graphs in the form of icons labeled with the letter E. An average enzyme activity is set by default. At the left click on the icon labeled as enzyme system, a window appears with the corresponding indicator of activity of the given enzyme. Let us check, for example, the activity of the glucose-6-phosphate isomerase enzyme. In the graph of glucose transformations, this enzyme is located between glucose-6-phosphate and fructose-6-phosphate. We can also follow the energy output as the result of metabolic reactions in the given organ. That's what the electron chain mode has been engaged for. Here we can find the ATP value changing in the real-time mode. A user can manually change quantitative indicators of activity of any enzyme and thus alter the course of any chosen biochemical process.